Hi, my name is Hasbani Edison, and I'm a master's student in computer science at UTRGV. My ongoing research with Dr. Ayadi focuses on kinase substrate associations. We're currently developing a precise method to identify kinase drug targets in complex diseases. This is the first step towards sustainable and effective treatment. A little background: kinases are enzymes that are important due to their role in several biological mechanisms. In particular, they play a crucial role in phosphorylation. Phosphorylation is when a kinase catalyzes transfer of a phosphate group to a protein, which then becomes known as the substrate of the kinase. This process is so critical that with any dysregulation of it, it causes a wide range of complex diseases such as diabetes, cardiovascular disorders, and cancer. There has been a movement from traditional drug discovery approaches to computational, and this is allowed by the available data that is mass spectrometry. There has been several computational methods that aim to identify the kinase substrate associations, such as Kinome Explorer. Kinome Explorer is a method that utilizes sequence match scoring and networking proximity to predict KSAs. It uses static information such as sequence motifs and physical interactions. On the other hand, CoPhos-K is a context-specific method that uses co-phosphorylation network to predict KSAs. It uses a Bayesian framework that utilizes multiple co-phosphorylation relationships of a phosphate to score and rank the top kinases of that particular phosphate. These methods, although good, they tend to favor well-known kinases. And by well-known, I mean kinases that have a lot of information on them already. The challenge here is that we need to identify the substrate of kinases that don't have as much information. Currently, less than 5% of the recorded phosphates have a known kinase. So our goal here with our research project is to develop a prediction method for these kinases with little information. Our prediction method will generate a network of kinases and substrates by using algorithms that were developed for gene regulatory networks, GRN. So NetRex is a method that was developed in this context, the gene regulatory networks, GRN. It finds the links between the transcript factors and genes. And using this concept, we're going to modify it and use it for finding the associations between kinases and their phosphates. Um, we will modify the method and create a network where the kinases and the phosphates are nodes, and the known associations between them are going to be represented by edges. And this could be shown in figure 2. This method uses a network component analysis model, NCA. And essentially, this one assumes that the kinase is characterized by its activity. We're going to use NCA to explain the phosphorylation of each phosphate as a linear combination of the activities of its regulating kinases. We're going to be adding and removing edges constantly to our network, and the method's gonna preference those that are co-phosphorylated sites and with kinases correlated activities to correlate. Sorry, the graph is gonna preference the topologies where kinases have correlated activities and they correlate the same site. So we're going to have one kinase with similar activities that's going to basically phosphorylate the same site. This method is formulated as a linear regression problem, and it models a relationship between the phosphorylation of phosphates and the unknown kinase activities of their regulators. So for our workflow, we collect the data. We're going to collect mass spectrometry data, which is essentially different samples of cancer, from breast cancer to ovarian. And we're gonna get the kinase substrate data, and we're gonna get this from the Phosphocyte Plus website. We'll then going to pre-process our data to transform it into acceptable input files for NetRx. We're gonna modify NetRx um, for it to work for kinase substrate predictions instead of GRN. And lastly, we're going to perform performance analysis and compare it to different other methods. We're going to use cross-validation to assess the sensitivity and specificity of our network to make sure that we're getting, reasonable results, and if we're actually calculating results for unknown kinases. So progress so far, we have the data collection completed. We are transforming the datasets to be acceptable input files. And we're also already working on modifying the algorithm to adopt it into a new model for our datasets. As I mentioned earlier in our introduction, um, the dysregulation of phosphorylation is what causes cancer. So with our research, we're really hoping to make a sustainable impact. We are contributing to Sustainable Development Goal number 3 from the list of 17 sustainable goals from the United Nations. And it's essentially trying to reduce by 2030 the premature mortality from non-communicable diseases, which includes cancer. So to be able to make a sustainable impact, we have to assess our research and how it's going to impact the 3 pillars, which are society, environment, and economy. For society, the current anti-drugs that are on the market, they kill cancer cells, but they also have an effect on healthy ones, and this causes numerous side effects that could be irreversible. So with our method, we're hoping to determine the best kinase to target for the treatment of cancer with decreased side effects, instead of kind of guessing what cells causes it, what cell they should attack. This would be a more precise treatment. For environment, research shows that there has been anti-cancer chemical compounds found in water, hospital and wastewater effluent, and groundwater and surface water such as lake, creeks, and ponds. And this could be a great risk for our aquatic environment. So this is a huge thing that we are working towards. Um, our method aids in the production of a specific treatment called kinase inhibitors. So what kinase inhibitors do, they locate a certain kinase, and they how can I say it? They put it to sleep, they stop it, um, with hopes of, you know, treating cancer. So these kinase inhibitors, they have been the least, research shows that they have been the least detected in the environment. So this could be a huge improvement from the current anti-cancer drugs out there. Economy-wise, cancer treatments are extremely expensive for patients, for insurance companies, basically for everyone. Our method contributes to the movement towards precision medicine, which is basically assessing a patient and creating a prescription specifically for them. This has the potential to reduce the treatment costs in the long term without using so many drugs and kind of guessing what drugs will be more effective. Um, in conclusion, um, we know that identifying kinases is a direct driver into finding cancer treatments, into drug discovery. And we need to develop computational methods that focus on predicting cases for kinases with unknown information. And our method creates a network of kinases and substrates to model KSA predictions and hopefully find unhidden kinase activities to be able to make predictions for these kinases with little information. We also want to acknowledge the Office of Sustainability for helping us with the sustainable portion of our research. Thank you.